Good day.
I am Joanna Margarito Pukol, a third year BS Biology student. So for today, I will gonna discuss about ectodermal placodes and the epidermis. So let's start. Chapter 16 entitled Ectodermal Placodes and the Epidermis. I will discuss first the cranial placodes, the senses of our heads, second, the dynamics of optic development, the vertebrate eye, third, the lens retina induction cascade, and lastly, the epidermis and its cutaneous appendages. Before I will discuss the main topic, I will first define the placodes. Placodes are a localized region of columnar epithelium that develop from ectoderm between the 10 to 30 somite stage at the border between neural plate and neural crest and the feature epidermis. Our organs of sight, hearing, smell, and even the hairs on our skin started as simple thickenings in the surface ectoderm called the placodes. These placodes and the skin's epidermis are formed from the non-neural ectoderm that remains external after the neural ectoderm has moved inward to create the central and peripheral nervous system. Non-neural ectodermal placodes are born through interaction with surrounding cells, the tissues of the endoderm, mesoderm, and neural plate, and the types of interaction differ depending on the placode. Next is the ectodermal placodes. It is one of the important type of epithelial reorganization. It is the thickenings of the surface ectoderm that become the rudiment of numerous organs. These include cranial sensory placodes, such as olfactory nasal, auditory ear, and lens or eye placodes, as well as the placodes that give rise to non-sensory cutaneous structure, such as hair, teeth, feathers, mammary, and sweat glands. So ectodermal placodes are focal thickenings of the cranial ectoderm that generate many different components of the sensory system of the head. The emergence and utilization of this embryonic structure have long been viewed as important for vertebrates' evolution. Among the features that distinguish the vertebrates from other chordates is the accumulation of these placodal-derived sensory structures in the head. And now, I will discuss the cranial placodes, the senses of our head. Cranial placodes. The vertebrate head has a concentration of neurons critical for sensation and perception. In addition to the brain, the eyes, nose, ears, and taste bud are all in the head. Head also has its own highly integrated nervous system for sensing pain and pleasure. For this sensing pain, let us think of the trigeminal nerve that innervates the teeth. And then this pleasure, think of the receptor on our lips and tongues. The element of this nervous system arise from the cranial sensory placodes, which are the local and transient thickenings of the ectoderm in the head and neck regions of the embryo. So these specified cells called cranial placodes. They are kind of the precursor to sensory neurons. And so, around the developing neural tube here, this neural tube, you see that there are these specified cells that are kind of anterior from the neural crest and sort of around the neural plate. So they're sort of around the neural plate. And these cells will eventually become the cranial placodes, which then go on to become sensory neurons. And so some examples are olfactory neurons. So these are they're tied to smell. You have lens cell. So obviously it has to do with vision. You also have the trigeminal cells, which will give rise to the trigeminal nerve, a sensory nerve associated with your face and mucosa in your mouth and the nasal cavity. You have geniculate. This is another nerve that is kind of in your ear region. And we also have this petrosal, which is another nerve that connects to the facial nerve as well as to the otic and the nodus, which is a jugular nerve. If you can see, this is the shape of the positioning up ahead if the head was kind of flattened outright. So we have the olfactory, which would be where your nose is, and then your eyes, and then your facial nerve, and then down at the bottom, one that's near your jugular. With some contribution from the cranial neural crest, the cranial placodes generate most of the sensory neurons of the head associated with hearing, balance, smell, taste, pain, temperature, and touch sensation, and even blood pressure. The cranial neural crest contribute to all of the glia and proximal portion of the sensory ganglia. So, the illustration is just the same that I have explained a while ago. So first is the anterior cranial placodes. Adenohypophyseal placode first developed into a structure known as Rothk's pouch before differentiating into the anterior lobe of the pituitary gland. So this is the adenohypophysis. Second is the lens placode. It invaginates to form the lens vesicle, which latter form the lens of the eye. The adenohypophyseal and lens placode are the only cranial placode that do not generate sensory neurons. All other placodes in the head do. Next is the olfactory placode. Give rise to the sensory neurons involved in smell, which is the first cranial nerve, the olfactory nerve, as well as a variety of migratory neurons that will travel into the brain, including but not limited to those that will secrete GDH or gonadotropin releasing hormone. Next is the intermediate cranial placodes. Trigeminal placode. These generate the distal neurons of the trigeminal ganglion, whereas the proximal neurons of this ganglion are formed from neural crest cells. So basically, it is divided into the ophthalmic and maxillomandibular placodes. And as I have said earlier, it generates the distal neurons of the trigeminal ganglion, trigeminal nerve, function to support the sensation of touch, temperature, and pain. So this is the trigeminal nerve. The otic placode give rise to the sensory epithelium of the inner ear and to the neurons that form the cochlear vestibular ganglion or the eighth cranial nerve, which are important for transmitting sound and balance information to the brain. Next is the epibranchial placodes. These are made up of the geniculate petrosal nodus placode, which give rise to the distal part of different cranial nerves, such as 7th, 9th, and 10th. The geniculate-derived nerve innervate taste bud, tonsils, and ear lobe. Next is the petrosal-derived nerve innervate the tongue and carotid sinus of the body. The nodus-derived vagus nerve innervate many other organs of the body, such as heart, lungs, and GIT, or gastrointestinal tract. So this picture shows the specification of neural crest cells. The neural plate is bordered by neural crest anteriorly and caudally, and by the placodal ectoderm anteriorly. So this is placodal ectoderm. The ectodermal cells receive both BMP and WNT for an extended period of time, they become epidermis. If WNT induces BMP and this is downregulated, the cells become placodal cells, or the expressing the placode specifier genes 61, 64, and AYA2. If WNT induces BMP but remains active, these border cells between the neural plate and epidermis become neural crest, which is the expressing the neural crest specifier genes PAX7, SNAIL2, and SOX9. If they receive WNT only, signal is black binogen or the FGF. So the result is the ectodermal cells become neural crest. Placodes are endorsed by their neighboring tissues, and there is evidence that the different placodes are each a small portion of what had earlier been a common pan-placodal field that is competent to form the placodes if endorsed. Cranial placodes. It may be induced from a common set of inductive signals arising from the pharyngeal endoderm and head mesoderm. So as you can see in the screen, this is a diagrammatic summary of some of the signaling pathway involved in the specification of the pan-placodal field. and the induction of cranial placode from this region. Here we see how these neural crest cells turn into tissues, and so we start with the expression of this FGF, which turn these neural crest cells into pre-neural cells. If the anti-BMP and anti-WNT are expressed, then those pre-neural cells become neural cells. This FGF and BMP are expressed, we get a state called border cells. And then, if BMP, WNT, and RA are expressed, we move balik to pre-neural cells. Ang balik diri sa pre-neural cells or these pre-neural cells. Again, and now, if anti-BMP and anti-WNT are expressed in the border cells, so after they have become border cells, which is conversed right here, conversed right here, then they will become this pan-placodal field. So they are again kind of this grouping of cells that are not yet specified. So once this pan-placodal field, cells express different genes. In this case, for example, this FGF2, 3, 8, and 19, that cells will develop into the olfactory tissue. And if that pan-placodal field expresses WNT and PDGF, we get trigeminal, and if it expresses FGF3 and 8, we get the epibranchial. And if this expresses WNT, FGF, and Notch, then we get the otic. Next is the induction of the otic placode in the cheek embryo. So the cheek otic placode, which develop into the sensory cells of the inner ear, is induced by a combination of FGF and WNT signaling. FGF19 from the underlying cranial paraxial mesoderm is received by both the presumptive otic vesicle and the adjacent neural plate. FGF19 induces the neural plate to secrete this WNTHC, and then FGF3, which in turn act synergistically to induce the formation of otic placode. So siya, and then they induce the formation of this otic placode. The localization of FGF19 to the specific region of the mesoderm is controlled by FGF8 secreted in the endodermal region beneath it. Let's look at the induction of the otic placode to become these inner ear nerves. So this is an example of the cheek embryo. And so what happens initially is that the pharyngeal endoderm, which is down here at the bottom, will express FGF8, which is a paracrine factor. And that FGF8 will find its way to the mesoderm tissue, which is indicated, mesoderm, this is indicated as pink label, and that will induce or lead to the expression of this FGF19. So this is the FGF8. So this FGF19 travels to both, the neural plate and the otic placode. Here in the neural plate, it will cause the expression of FGF3 and WNT8C, which is then, that signal is sent to the otic. This otic placode, and then with the help of this FGF19 that we saw living earlier in the mesoderm previously, it will lead to the expression, wala siya nabutan dito, so it led to the expression of PAX2, which is, as what I have said earlier, which isn't labeled here. So PAX2 will start this cascade of the other genes that will allow the specification and production of the otic, that they will then move on to become the sensory cells, a finalized sensory cell. Next, we will discuss the dynamics of optic development in the vertebrate eye. The most basic part of the embryonic vertebrate eye include the retinal pigmented epithelium and the neural retina and the lens. While the retinal pigmented epithelium and the neural retina arise from the central nervous system, or the lens, which start out as a lens placode, is derived from the non-neural ectoderm. Unlike most other sensory placode, the lens placode does not form neurons. Rather, it forms the transparent lens that focuses the incoming light onto the neural retina. The retina develops from the optic vesicle that forms a lateral bulge of the diencephalon, a region of the forebrain. The interactions between the cells of the lens placode and the presumptive retina structure the eye via a cascade of reciprocal signaling events that enable the construction of the intricately complex organ. Next is the vertebrate eye. So during gastrulation, the involuting precordial plate and the foregut endoderm interact with the overlying precaudal region, inducing it to have an anterior character and conferring upon it a lens-forming bias. These tissues induce a number of anterior genes, include the PAC6, which code for the transcription factor, this PAC6, which encodes transcription factor in the ectoderm, which is critical in the competence of the ectoderm to respond to subsequent signals. Interestingly, the entire preplacoidal region is initially specified as a lens. Even cells that normally form the ear or form the ear or other placode, since not all parts of the preplacoidal region eventually form lenses. Lens must have a precise spatial relationship with the retina. The repression of lens potential is achieved by migrating NCC, which is the neural crest cells. These neural crest cells are blocked from making contact with the pre-lens region by outgrowing optic vesicle. This, along with signals from the optic vesicle, position the lens in relation to the retina. Next, development and reciprocal induction of the vertebrate eye. So this picture shows the development and reciprocal induction of the vertebrate eye. So in picture A, it's with the optic vesicle invaginating and contacting the overlain ectoderm, which induces the lens placode. Mo-induce siya lens placode. And from this point, the overlaying ectoderm differentiate into lens cells. It differentiates into the lens cells, and the optic vesicle folds into itself. Kung kaning optic vesicle mo-fold into itself lang siya. So you can see that it is kind of smashing down right there. And at that point, the previous optic vesicle, which was folding in itself, finishes folding to form this optic cap. So pag human aning optic vesicle nga yang fold iya ang kaugalingon, it will form to the optic cap and will become the neural and pigmented retina. And the lens in turn, which was the lens placode, it will internalize. So you can see kind of a broken off from being part of the external layer of the scene, and it's folding in and being internalized. And now that the lens is internalized, this is another case of reciprocal induction. So these cells have specified to become lens cells, and they have been internalized and have started to form the lens structure, and they will induce these outer ectoderm cells here to become the cornea. Formation of the eye field, the beginning of the retina. The details of eye development tells us how eyes comes to be made only in the head and why only two eyes normally form. These details show that the precise arrangement of the eyes is the result of the multiple layers of inductive event involving gene expression, differences in both time and place. So the story begins with the formation of the eye field in the anterior of the neural tube. The anterior portion of the neural tube, where both BMP and WNT pathways are inhibited, specified by OTX2 gene expression. Noggin is especially important because as it is not only it blocks BMP and and inhibit the expression of the transcription factor ET. However, once OTX2 protein accumulate in the ventral head region, it blocks the Nogin's ability to inhibit the ET gene, and then kaning ET protein mo siya mahimong maproduce. One of the genes controlled by ET is this RX, whose product helps specify the retina. RX meaning for retinal homeobax. It is a transcription factor that acts first by inhibiting the OTX2 since OTX2 has finished its job and now might interfere, and second by activating PAC6, the major gene forming the eye field in the anterior neural plate. So this PAC6 protein is especially important in specifying the lens on retina. It appears to be a common denominator for specifying photoreceptive cells in all phyla, including the vertebrates and the invertebrate. Next is the dynamic formation of the eye field in the anterior neural plate. So let's look at some of the genes involved in this process of the eye development in the eye field. So we start off with this light blue light area and the light blue. In this case, it represents the neural plate. And so, the nagin causes the induction of formation of the neural plate. When siya nagin it causes the induction of formation of the neural plate. And then this OTX2 will be expressed only in a certain area of the neural plate. And that leads to the development of the forebrain or midbrain. And we still have the remainder plate behind here. Then we have the expression of ET, RX1, PAC6, and 6-3, as well as the LHX2. And then two or more genes come in. So, moapil ang TLL and OPTX2 that will further lead to the cascade to the full development of the eye from the eye field. So while we listed all these transcription factors as playing a role in eye development, they're not all active across the entire developing eye field. So starting with 6-3, it covers the large path of the eye field, or it covers the largest amount of area. Next is we get more and more specific. So 6-3 covers the large path as what I have said earlier. Then this PAC6, it is a little less. RX1, a little less than that. LX2, less than that. And then finally, the ET is the most specific. And so this kind of lead into the ideas that these different gradients, right? And so you don't want all genes to express all over the place because you need to specify. So especially that in the eye where this very specific tissues, rod and cons, lens, and also the cornea that are all very tightly packed together but all very different. And so those overlapping expression where we get from general to more and more specific, it helps in that to aid the development specificity of these cells in the developing eye field. Next is the dynamic formation of the eye field in the anterior neural plate. So humans and mice heterozygous for loss of function mutation in PAC6 have small eyes, whereas homozygotic mice and humans lack eyes altogether, as do RX mutant mice. In both plies and vertebrates, this PAC6 protein initiate a cascade of transcription factor with overlapping function. These factors mutually activate one another to generate a single eye forming field in the center of the ventral forebrain. The final result is two eyes that are more lateral in the head, and then the main player in separating the single vertebrate eye field into bilateral field is our old friend, which is the SHH or sonic hedgehog. So I have an explanation about this picture. So as you can see, this is the normal and the RX knockout. So as shown in the picture, this is the example of a mouse that has RX knockout. Then you stop the development of that organ. So just like any other organ that we talked about previously, these genes are vital for this cascade because these transcription factors play a role to express other genes that led to further development. So here in the picture, the knocking out of X in this mouse leads to no development of an eye. Also, the hedgehog from the precordial plate suppresses PAX6 expression in the center of the neural tube, dividing the field in two. If the mouse SHS gene is mutated or the processing of this protein is inhibited, the single median eye field does not split. So the result is cyclopia, a single eye in the center of the face and usually below the nose. These sonic hedgehogs separate the eye field into bilateral field. Gervine, which is an alkaloid found in certain plants, inhibit the endogenous SHH signaling. So first, I will discuss this A. Scanning electron micrograph showing the external facial features of a normal mouse embryo. This letter B is the mouse embryo exposed to 10 Gervine had variable loss of midline tissue and resulting fusion of the paired lateral olfactory processes or the OLF, optic vesicles or the OPT, and then the MX or the maxillary, and then the MN, which is the mandibular or mandibular process of the jaw. This letter C is the complete fusion of the mouse optic vesicle and lenses. So this is the optic vesicle and this is the lenses, resulted in the cyclopia. This letter D naman, it shows the drawing the location of the precordial plate, which is the source of sonic hedgehog in the 12-day mouse embryo. Conversely, if too much sonic hedgehog is synthesized by the precordial plate, the PAX6 gene is suppressed in too large an area, and the eyes fail to form at all. This phenomenon may explain why these cave-dwelling fish are blind. Cave-dwelling fish. As you can see in the picture, blind siya. Wa siya mata yan. Siya mata but it's blind. So the difference between the surface population, while this surface population of the Mexican tetrafish, or in scientific name, Astyanax mexicanus, and the eyeless cave-dwelling population of the same species is the amount of sonic hedgehog from the precordial plate. Ang hinungdan ani kay parehas na siya nga kuan. They are the same fish or the both the Astyanax mexicanus and the cave-dwelling fish or cave-dwelling population. Same species lang sila. But ang naka-problema ani kay kaning amount of SHH or the sonic hedgehog from the precordial plate. Maong this cave-dwelling population is eyeless or blind. Elevated sonic hedgehog was probably selected in cave-dwelling species because it resulted in heightened oral sensing and larger jaws, yes. However, this sonic hedgehog, also downregulate the PAX6, resulting in the disruption of the optic cap development or the apoptosis of lens cell and arrested eye development. So I have this an explanation or clear explanation of this picture. So the surface dwelling population, this letter B, which is the cave dwelling population, or the Mexican tetras or Astyanac mexicanus. The eye fills the form in the population that has lived in cave for more than 10,000 years ago. So two genes, there are two genes, the PTC2, PTC2 and the PAX2, respond to sonic hedgehog and are expressed in broader domains in the cavefish embryo than in those surface dwellers. Ionic optic vesicle, which is in the bottom of the surface dwelling fish, are normal size and have small domains of PAX2 expression. Kaning PAX2 mo na siya, this is the specifying optic stalk, where this ang kanina namang paxic, which is it is usually expressed when the optic vesicles of the cave dwelling fish's embryo are much smaller. So kaning PAX2 expressing region has grown at the expense grown siya because of the expense of the PAX6 region. Lens retina induction cascade. Modern studies of vertebrate eye formation were initiated by Hans Spemann, which is in 1901, who found that when he destroyed the anterior neural plate on one side of the embryo, no lens formed on the affected side. While this Warren Lewis, which is 1904, found that when he placed an anterior neural tube under a different part of the head epidermis, the neural tube became a retina and the skin became a lens. More recent studies have shown that although the story is more complicated, the eye field of the anterior neural plate induces the epidermis to become the lens. As each lens begins to form, it induces the eye field to become the retina. The development of the eye is a beautiful example of reciprocal embryonic induction. So this picture shows the reciprocal embryonic interaction between the developing lens placode and the optic vesicle from the brain. So before I will discuss this, I will first read this one.
Starting very early during gastrulation and neural plate formation. So as you can see, we have the neural plate and the presumptive retina starting to develop here on the neural plate. We also have the lens ectoderm here that it's going to interact with. And as we progress through, the mesoderm here is going to induce the start of this formation of the eye.
Wait. Balik. So, nag-start ang embryonic interaction of the development of the lens placode and then the formation of the optic vesicle. So, we're starting very early during gastrulation. Masya nag-start diri sa gastrulation and neural plate formation. So, as you can see, we have this neural plate, which means NP, man siya NP, neural plate, and the presumptive retina, PR, they start to develop here. So, nag-start sila develop while napa sa neural plate. So, we also have the lens ectoderm, the presumptive lens ectoderm, which is the PLE, muna siya lens ectoderm. That is, muna siya murag mo interact with and as progress through the mesoderm, there is going to induce that start of this formation of the eye. So, kanang optic vesicle, ang optic vesicle will become, ang optic vesicle mo siya murag mo bulge type, like mo bulge siya, then and then it will contact the ectoderm. Katong mo bulge mo siya mo contact sa ectoderm, and at this point, this kind of start the cascade mo siya mo start na sa cascade of interaction that mo siya mo turn into the optic vesicle. So let's start.
Mesoderm here is going to induce the start of this formation of the eye. And so the optic vesicle will become a bulge. Where's my marker? Here we go. And it will contact that ectoderm. And at this point, this kind of starts the cascade of interactions that turns this optic vesicle. It'll start to fold in on itself, and eventually it will become the optic cup. And so at this point, before it becomes the optic cup, this connection between the optic vesicle and the lens placode cells here, the light blue, this is where that reciprocal induction occurs. And so here you can see that the optic vesicle is going to transform to the neural retina.
The video. Mo na siya mo contact kani. Siya contact sa ectoderm. Mo siya mga bulge, katong bulge mo contact. Mo to mo forming siya optic vesicle. Sa ganito mo form. So kani wait ha. So di ba may ko ganina nga katong nagkontak katong nang bulging mesoderm do ya. The katong mga optic vesicle mo na siya mga optic vesicle mo contact siya to the ectoderm to kind of start the cascade of interaction that turn this optic vesicle. So as you can see, it will start to fold na siya in on itself, and eventually it will become optic cup. Form siya OC or the OT, a optic cup, this OC. Mani siya OV, she's the bulging. Mo-contact siya sa mesoderm. So you can see the kani OV, contact niya siya sa mesoderm or the LP, which is the LP, siya lens placode. Yung contact na siya, and then it eventually it form into the, kabantay mo pag-abot diri nga picture, para siya hingtapot pag-ayo, and then murag siya na siya cascade of interaction nga kaning optic vesicle maghinahina siya fold into itself, and eventually it become this mohimo na siya OC, di na siya baka nag-OV.
The lens placodes, which will then become presumptive lens cells. Then those cells will reciprocate, and they will send back FGF for these optic vesicle cells to then develop into the neural retina. Now, once that occurs, we get the full folding of this optic cup, and it'll convert the inner layer into the neural retina, and it will cause the lens vesicle to invaginate or involute and form the lens vesicle. So that's where those the presumptive lens separate, and the endoderm then covers, which will eventually become the cornea of the developing eye.
Siya LP, lens placode, nagakuan na siya. Mo siya mga kaning lens placode, mo siya light blue. This is where that reciprocal induction occur. So makita nimo nga ang optic vesicle is going to the transform to the natural retina, and that is going to send this orange dot na siya transform to the neural retina, and then it is going to send siya mga orange dot mo to mga FGF. Yang send These orange signaling factors, which are the one siya FGF8, BMP4, and then the delta. Delta to the lens placode, which will then become a presumptive lens cell. So, as what said this while ago, ingon niya nga, ning FGF8, katong mga orange nga dot, orange signaling factors, and BMP4 and delta cells, and delta the lens will send back, mo siya, murag siya ba exchange, will send back FGF. For this optic vesicle cells to then develop, mo siya, develop into siya sa neural retina, mo siya NR, neural retina. Now, once that occurs, we get the full folding of this optic cap vesicle, folding of this optic cap, and it will convert the inner layer into the retina. And it will cause the lens vesicle to invaginate or involute the lens vesicle. So, nakagets lang mo. At this point, it becomes a Judiciag optic cap. This connection between the optic cap and the between the optic vesicle and the lens placode cells here at the light blue siya, is where that reciprocal induction occurs. Muna siya ngayon nga, na siya form na siya induction occurs, or the reciprocal induction. So, kabantay mo diri nga kuan ay mga gagmay nga dot, kaning kuan, kaning lens placode, hinahinay sila send og signaling factors mo ng mga FGF8, BMP4, and delta to the lens will send back, mo send back para siya sa speed FGF. For this optic vesicle to then develop, para ma-develop siya into the NR or the neural retina, para ma-develop siya NR or neural retina. Lens and cornea differentiation. The differentiation of lens tissue, a transparent membrane capable of directing light onto the retina, involves changes in cell structure and shape, as well as the synthesis of transparent lens-specific proteins called the crystallines. These crystallines account for up to 90% of the lens-labeled proteins. The lens cell must curve properly, and this curvature is caused by balancing the raw generated apical construction microfilaments with rock-generated actin polymerization that extend the microfilament along the apical basal axis. So the lens crystalline containing posterior primary fiber cells eventually elongate and fill the lumen of the lens vesicle. The anterior cells of the lens vesicle constitute a germinal epithelium which continues dividing. These dividing cells move toward the equator of the vesicle, and as they pass through the equatorial region, they too begin to elongate into secondary cellular fibers. With maturation, these fiber cells, lose their cellular organelles and their nuclei are degraded. Thus, the lens contains three regions, known as the anterior zone of dividing epithelial cells, an equatorial zone of cellular elongation, and lastly, a posterior and central zone of crystalline containing fiber cells. So as you can see in the picture, this is the differentiation of lens on anterior portion of the mouse eye. So at first, at embryonic day 13, the lens vesicle detaches from the surface ectoderm and invaginate into the optic cap. Corneal precursor. These corneal precursor cells, or the mesenchymal cells, from the neural crest migrate into this space. Hinahinay siya migrate, and then migrate. The elongation of the interior lens cell begin and producing primary lens fiber. Hinahinay siya produce a primary lens fiber. At the letter B, which is the day 14, the lens is filled with crystalline synthesizing fibers. Puno na siya crystalline synthesizing fibers. Then the neural crest derive from mesenchymal cells between the lens and the surface, condense to form para ma-form og several layers. Kaning letter C naman, it is at day 15. Kanya kay day 13 siya, day 13, 14, then day 15. At day 15, ang lens kahinay-hinay detaches. Ito yung mga lens, hinay siya detaches, di ba? Kasi kanagay na color kana siya purple ba na purple. Hinay siya detaches until pag-abot sa day 15 ni-detach na jud siya from the corneal layers, generating an anterior cavity. While in letter D, the surface ectoderm at the anterior side becomes the corneal epithelium. And at day 15.5, corneal layers differentiate and begin to become transparent. Hinahinay na siya transparent. The anterior edge of the optic cup enlarges to form a non-neural region containing the iris muscle and the ciliary body. New lens cells are derived from the anterior lens epithelium. As the lens grow, the nuclei or the primary lens degenerate and new lens fibers grow from the epithelium on the lateral side. Paracrine factors from the optic vesicle also induce lens-specific transcription factors. Regulation of the crystalline gene is under the control of PAC6, SOX2, and the IMAF. Like OTX2, PAC6 appears in the head ectoderm before the lens is formed, while the SOX2 is induced in the lens placode by BMP4 secreted from the optic vesicle. Co-expression of PAC6 and SOX2 in the same cells initiate lens differentiation and activate crystalline genes. So appearing later than SOX2 is the IMAF. Later after the SOX2. It is induced by the FGF8 secreted by the FGF8 secreted from the optic vesicle. Co-expression or the IMAF is induced by the FGF secreted by optic vesicle and is needed. They are needed for the maintenance of crystalline gene expression and the computation of lens fiber differentiation. So as you can see in this letter E, it is a close binding of the SOX2 and the PAC6 transcription factors on a small region of the crystalline enhancer. The last topic is the epidermis and the cutaneous appendages. The skin, a tough, elastic, water-impermeable membrane, is the largest organ in our bodies. Mammalian skin has three major components. First is the stratified epidermis. Second is the underlying dermis composed of that loosely packed fibroblast. And third is the neural crest, which derive from the melanocytes that reside in the basal epidermis and hair follicles. So it is the melanocyte that provide the skin pigmentation. In addition, a subcutaneous or below the skin fat layer is present beneath the dermis. Moreover, skin is constantly being renewed, and this regenerative ability is possible due to the population of epidermal stem cells that last a lifetime. So the epidermis generate from the ectodermal cells covering the embryo after neurulation. This surface ectoderm is induced to form epidermis rather than neural tissue by the actions of BMPs. These BMPs promote epidermal specification, and at the same time, it induces transcription factors that block the neural pathway. So the continuation of the origin of the epidermis. The epidermis is only one cell layer thick to start with, but in most vertebrates, it soon becomes a two-layered structure. The outer layer gives rise to the periderm, a temporary covering that is shed once layer differentiate to form a true epidermis. The inner layer called the basal layer, this basal layer or the stratum germinativum or stratum corneum that contains epidermal stem cells that attach to basal lamina the stem cells themselves help to make. Just about all the major signaling pathways are involved in the forming ectodermal appendages. In some instances, such as the enamel knot of mammalian teeth, the same signaling center sends out paracrine factors belonging to nearly every family. The canonical WNT or the beta-catenin pathway was implicated by the loss of property formed here, property formed, not proper like properly formed, such as hair, teeth, and mammary glands in mice deficient in components of this pathway. Manipulating embryos to have beta-catenin expression throughout the epidermis eventually transforms the entire epidermis into a hair follicle fate, and it creates supernumerary teeth in the jaw. WNT signaling may help induce the enamel knot to form and may be critical in the ability to regenerate teeth. So mouse mutant lacking negative regulators of WNT pathway display more and larger mammary placodes. A continuation of signaling pathway. If GF regulate the migration of mesenchymal cells to become the condensate beneath the placode. So we all know that if GF probably play multiple roles in ectodermal appendage development. So one of these, as I have mentioned, is it regulate the placode to the migration of mesenchymal cells to become the condensate beneath the placode. So in hair, the placodal WNT activates, in teeth, if GF8 from the placode appears to attract and sustain the mesenchymal cells to the tooth placode. In hair, the placodal WNT activates the secretion of FGF20, which may stimulate mesenchymal cell migration to the placode. This FGF10 from the somite in the mammary gland is that it's thought to induce placode formation. BMPs play an important role in ectodermal appendages formation. They are known to induce several genes involved in tooth development. While these BMPs and WNTs most likely regulate each other to control the tooth shape. While BMP expression is necessary for tooth formation, BMP activity must be suppressed for the induction of the hair placodes. Other signaling pathways, such as those initiated by hedgehog and ectodysplasine pathway, are also important to vary degree. This ectodysplasine pathway is active in every cutaneous appendage. The other animals with anhydrotic ectodermal dysplasia have defective growth of hair, teeth, and sweat glands. Anhydrotic ectodermal dysplasia. Lastly, the ectodermal appendage stem cells. This is the mammary gland that contains stem cells for the reactivation of its growth at puberty and pregnancy. During puberty, estrogen causes extensive branching of the ducts and elongation of the terminal end buds. During pregnancy, the ducts stimulated by progesterone and prolactin form tertiary side branches that differentiate into the milk-producing alveoli. The mammalian mammary gland probably contains a stem cell that is able to generate all the lineage of the gland. Data from genetically labeled mammy cells suggests that there may be a single stem cell that can make two major progenitor cells of the breast, one that generates the duct and alveoli, and one that generates the myoepithelial cells that contract to push the milk out of the alveoli and toward the nipple. So I will discuss this picture. So let's look at the breast development and female. And so breasts start to develop at embryonic day 11 in mammals or in mice. In this case, what happens is they go through this embryonic stage, and then at birth, they have kind of infant stage, and they will remain in that infant or newborn stage until mukha siya puberty, where kaning estrogen is expressed, and we start to see more and more development in the ducts. So expanding the ducts of those mammary glands, and then once you become an adult, progesterone during pregnancy, duct stimulated by progesterone prolactin. So this progesterone, which is kind of the finalizes the this enlarges this duct and proliferates this duct, and then this estrogen and progesterone and prolactins are expressed. They're both expressed during pregnancy. And as you start to get the alveoli, alveoli that are filled with the whole milk, alveoli ni mo na siya whole milk, and the afterbirth prolactin are expressed or right near birth in some cases. So mo siya led to the formation of the milk casein expression that will fill the alveoli with milk, and sacs will be filled filled with milk. But then after a certain amount, and after this certain amount of time, once breastfeeding is stopped, as soon as the breastfeeding stimulation of the nipple and of the breast stop, then also kaning lactation will stop sa niya. Pretty soon, as you stop stimulating the breast with the child feeding on it, or with pumping ng alveoli, will go through the apoptosis. Na siya apoptosis. It will go back to this adult state where it is not producing milk anymore, and then subsequently, if you have a wife or anybody wants to get pregnant a second time, they go through the cycle again. Pag manganak ka, balik po ka mo process ka estrogen, progesterone, prolactin, then if na pregnant na ka na kay prolactin, which this turn into the lactation sa imong alveoli. So magbalik-balik ra gyapon iyang cycle until di na ka manganak. So it can happen over and over and over again. So I hope that you learned something from my report or my video. So thank you so much. God bless. Bye.